Let's take a quick look at metabolism in Chapter 2, and in particular, digestion. The beginning of each chapter does have a question to pose. You are the coach. What kind of question? And then there are typically 2 to 4 questions that go along with each little case. So something to think about would be when you are done with the reading, go back and take a look at this and see if you can apply the content. The purpose of digestion is to break food down into components that can be absorbed or assimilated and used for energy. And as you remember, there are micronutrients and macronutrients, which would be grams versus micrograms. And each part of the tract has a function, and the nutrients are absorbed at different times throughout the digestive tract. The components of the digestive tract are shown in Figure 2.1. The salivary glands, when you first put food in your mouth, are going to secrete an amylase, which begins to break down starchy foods into glucose molecules. Digestion in the stomach is involving really a preparation for further breakdown, preparation for absorption into the small intestine, and in some regard primarily the small intestine. The HCl or hydrochloric acid in the stomach denatures amylase and halts carbohydrate digestion at that time. The mechanical component of moving that food around, it forms what you probably recall from anatomy, a bolus that moves into the small intestine. The only carbohydrate that is absorbed in the stomach is alcohol. The small intestine involves cooperation from the liver, which is going to release components, bile, which is going to emulsify fats, the pancreas, which is going to help with carbohydrate absorption, and also farther down you'll see the components of the length of the components of the digestive tract and what happens at each one. So Figure 2.4 is a nice little review. The small intestine is where a lot of absorption occurs, and it has a very large surface area based upon its finger-like projections, the villi, and then on each one of those villi there is even smaller projections and little brush border. That microvilla and villa really increase the surface area for your digestion. It's similar to the alveoli in the lung. It increases surface area of oxygen exchange, while the microvilla and the villa are perfect for increasing absorption for fat, proteins, and carbohydrates. The large intestine has a small amount of absorption, and you'll want to remember water, potassium, sodium, vitamin K, and chloride. Most of the minerals, vitamins and minerals, are absorbed in the small intestine, but those are the exception. The large intestine has a little bacteria component which helps to break down fiber and other digested carbohydrates, produces gas, absorbs the water, and gets the feces ready for excretion. What you'll want to do in Figure 2.7 is just take a look and see what is occurring at different components. You'll see that alcohol is absorbed in the stomach directly into the bloodstream. As you go down the small intestine, you'll see water, minerals, and carbohydrates are being absorbed. As you get farther down the small intestine, carbohydrates, water, that's my dog, hello Cosette, and water and vitamin K back in the large intestine. Absorption occurs through four primary mechanisms. One is passive diffusion. Passive diffusion is a substance that easily moves in and out of the across the cell membrane. It's either through protein channels, which you'll see are the purple components, or directly through the cell membrane itself. You can see that water-soluble substances, small lipids will move, and they'll go from a high concentrated gradient outside of the cell to a low concentration gradient inside the cell. Facilitated diffusion means that there has to be some facilitation or some assistance that needs to occur for substances to be able to get inside the cell. That requires a protein membrane, and this, for example, would be the way that fructose gets into the cell. The protein binds to the substance, and it changes shapes, allows the substance to move from a high concentrating gradient to a low concentration gradient and move into the cell. It's both passive and facilitated diffusion, high concentration to a low concentration. But with facilitated, it just needs to have some assistance through that protein binding site. Active transport occurs when there is a movement against a concentration gradient. So that is when there is a requirement of ATP or energy to be able to move into the cell. So the active transport is movement against, it's like swimming upstream, takes extra energy in the form of ATP. Endocytosis is when a substance uses, or the cell membrane engulfs around that particle and brings it inside the cell. That engulfing component portion of the membrane separates and encases that particle and moves it inside of the cell. So those are the four absorption mechanisms. This just shows you a little bit about carbohydrates itself. It starts by breaking down in the mouth. Amylase breaks down as an enzyme that starts to break down that carbohydrate. In the stomach, the acid stops that amylase, that salivary amylase, gets it ready to move toward the small intestine, and you have the small intestine, which is going to be primarily pancreatic amylase, which gets involved, and in the large intestine, bacteria are incorporate or act. Most absorption occurs in the jejunum in the small intestine, and it's primarily facilitated diffusion and active transport. After digestion, carbohydrates have some different directions where it can go. So you'll see that the broken down components of primarily carbohydrates are transported via the blood, and then there is cellular uptake by either insulin or glucose transporters. And the glucose transporters have specific names depending on where they're located. And you've got the liver, where glucose can be converted into either ultimately storage for fatty acids or in triglycerides for storage into a fat cell. You have glucose that can be stored in the liver as glycogen. You have glucose that can be released to almost all tissues, and particularly the working tissues, to generate energy via energy production. You have glucose that can be directly converted into triglycerides at the adipose cell or the adipose tissue. And you also have glucose going to the muscle, where it can be used for fuel or it can be stored as glycogen. So there are many ways, many places where glucose can go. It can be stored in the liver, converted to triglycerides at the liver. It can go to the adipose tissue. It can be used for nearly almost all tissues for energy, and also used at a muscle for work. The digestion of fat involves the enzymes named as lipases. So lingual means mouth, gastric refers to the GI tract, pancreatic means that the lipase has been secreted from the pancreas. And you'll see that the production or the breakdown of fats becomes progressively smaller individual pieces. It can be packaged ultimately in the small intestine to form micelles and travel forward. Very little fat makes it to the large intestine unless you have some sort of absorption disease such as steatorrhea. Fat primarily travels through the blood in small little packages. It's either carried with proteins like chylomicrons. It is taken up by cells by lipoprotein lipase on a capillary wall to break it down into free fatty acids and glycerol, or it can travel through the lymph. Main function of fat is for storage, involving storing of fat-soluble vitamins, protection, and also an energy storage, and also for use of energy. Protein digestion begins primarily, it starts in the mouth where you start to masticate or chew. The stomach churns. It causes denaturation of the amino acid structure, so it becomes more of a string. As it moves through the small intestine, proteases and peptidases are going to chop it up into smaller pieces, and then as it moves into the small intestine, it gets smaller. It ultimately is absorbed as amino acids. Proteins are absorbed in the small intestine, and the action is primarily facilitated diffusion, active transport, and very, very little makes it to the large intestine. After the protein is absorbed, it is transported through the blood, and it becomes part of the amino acid pool. The amino acid pool is necessary for the synthesis of new proteins. It's just a very small pool. It needs to be replenished through dietary consumption regularly. We also need to make sure that we have a good supply of good quality protein for the athlete. Protein is necessary for, or actually I should say the amino acids are necessary for the production of new proteins. If you were to take the exercise biochemistry class, you'd spend some time looking at the steps of transcription and translation. But the function of protein synthesis is to build or manufacture new proteins, and it requires good quality protein, essential and non-essential amino acids. So the bottom line is that good quality protein is necessary for the athlete's diet. And we also need to have sufficient protein. We'll eventually talk about what is sufficient protein. How much is it? That comes up in the future. And we'll look forward to talking about metabolism in just a moment.